So moving on with our discussion of the membrane, we talked about the overall structure, and now we're going to get into the physiology, the how it works. Whenever we are discussing living organisms, anatomy is the term that we use to describe the structure, and physiology is the term that we use to describe the function. So how does it work? And those go very hand in hand as we've been discussing. So here's some good learning objectives for this particular lesson. And let's dive in. So as we discussed, the orientation of these phospholipids is the key to the overall function of the membrane. Remember, the main job of the membrane is to control what goes in and out of the cell. And it's all because of how these phospholipids arrange themselves in this bilayer. It makes it selectively permeable, where we can control what goes in and out of the cell. That's all that selective permeability means. Selective means it chooses what particular things to let in and to keep out. Permeability is passing through something. So anything that is nonpolar and very, very small can cross the membrane directly because it's able to cross through this very nonpolar hydrophobic area, because either it's the same or it's small enough to not really cause a problem for the cell. Anything that is polar, anything that has a direct charge, because remember, polar molecules technically aren't charged. They're neutral overall. They just have kind of positive and kind of negative areas. So anything that has a charge, like an ion, sodium Na+, chlorine Cl-, those have direct charges on them. Those are going to behave similarly. They're very hydrophilic. And then we have large compounds. If we tried to poke something through here that was really large, it would kind of be the same as if you tried to put, what's the word I'm looking through? Like a meat cleaver through a balloon. It's going to cause massive problems for your poor little balloon. So anything very large, we have to move it across the membrane in a very specific way to avoid damaging the membrane itself. So anytime that we want to move something that's polar or charged across the membrane, that's where we need some of those transport proteins. They act as channels that allow water and all of its associated dissolved substances, like these hydrophilic charged polar things, to pass through. So before we get too further into this discussion of what's moving across the membrane and how, we have to understand the second law of thermodynamics. Now, we're not going to understand it in the sense of your engineering classes. Those are going to get significantly more detailed into this. We are going to look at it from a very broad lens and see how we can apply it to this particular biological situation. So the second law of thermodynamics states that the universe tends toward disorder, or entropy. Entropy will always win out. What does that really mean? Let me give you two examples. In the case of a person with a big stack of bricks, what are the odds that you could take a huge stack of bricks in your hands and throw them randomly and they would form a perfect wall? Highly, highly improbable. Impossible, there's always that tiny little fraction of a possibility, but really this is impossible to have happen. And that's because this is highly ordered. This is not following the second law of thermodynamics, where it says the universe is tending towards disorder. It's trending that way. What most likely happens is disorder occurs. The bricks that were in your hands fall randomly into a disordered chaos. So the same thing in a container. Let's take a moment and think about the atmosphere. If you don't know, the atmosphere is full of many different gases. Obviously oxygen, but the primary gas in the atmosphere is nitrogen. There's also carbon dioxide. There's also methane. There's also a few noble gases. There's lots of different overall gases that make up our atmosphere. Do they arrange themselves in a highly ordered way where all the oxygen is in one layer, all the nitrogen is in a different layer? No. If it did that, we'd all be dead. What really happens is they form as disordered of a situation as possible. So they will all randomly assort themselves into a chaotic mess. We end up calling this equilibrium, where they kind of spread themselves out equally everywhere within the atmosphere or within the container. So the idea is molecules are going to have a tendency to move from a more ordered state towards a more disordered state without any energy input, because this just follows the second law of thermodynamics. As these molecules are randomly vibrating, they are going to vibrate into a disordered state. This is what we call spontaneous. It happens without any external application of energy. If I wanted to put all the molecules in the jar like this, or if I wanted to make the bricks fall into a perfect wall, I would have to put in significant energy. And that's why these states don't really occur naturally, because you have to put in energy to make them happen. And that's not something that really, that's not the way the universe works. The universe will just do things based on likelihood and least amount of resistance, least amount of energy required, unless some external force comes in and says, no, I'm going to put in the energy and change this. So molecules naturally move from an area of high concentration, now concentration just means amount, to an area of low concentration. So let's look at this in terms of our particular example of the jar here. The blue molecules are in a highly concentrated area here because we have this entire upper portion that only has blue. There are no blue molecules on the bottom of this jar, so they are going to naturally want to move from where there is a lot to where there is few. And so they will naturally move downwards, and these yellow ones will naturally move upwards, just following that second law of thermodynamics. So this gives us the idea of a concentration gradient. So concentration just means amount. A gradient is where you have more of it in one area and then it slowly fades until there's less of it in another area. And these are so crucial to life. Cells will actually put in a significant amount of energy and effort to create these concentration gradients. Think of them as like a dam. If we can push all of our molecules over to one area and keep them there until we're ready, we've just stored up all this potential energy. And when we open up the floodgates and all of these molecules go rushing down, we can capitalize on that energy. So overall, concentration gradient means there's different amount of particles in one area compared to another. For the case of what we're discussing, most of the time imagine a membrane right here. So we're going to have a high amount on one side of the membrane and a lower amount on the other side of the membrane. Now, particles will move down their concentration gradient from a high to a low concentration just based on the second law of thermodynamics. And they will do this until they reach equilibrium, where there's a state of balance, or where the particles are equal on both sides of the membrane. So here we see a gradient. Here we have equilibrium, where it is equal chaos everywhere. Now, when we're talking about the cell and these concentration gradients, this is where it gets crucial. Things move with or against their gradient. They can either go down that gradient from high to low, or they can go from low to high. But that would require the input of energy. That would be like trying to put all of the oxygen in the atmosphere just in one little country, and then put all the nitrogen in another country, something like that. So passive transport inside a cell is where any movement follows the second law of thermodynamics. That's why it's passive. It does not require energy. Anything that is considered active transport is where you are attempting to create more order out of the chaos. You're not letting chaos reign. You're fighting it. So we're usually moving things against their concentration gradient from low to high. So we're in opposition to the second law of thermodynamics. Whenever that happens, we have to input energy. So it's going to require usually ATP, which is the cell's energy molecule, and we'll talk about that later. But there's many other ways that the cell can use energy to do active transport, and we'll talk about that too later. So passive transport would be like falling down a hill. Once you get started, you do not have to put any extra effort into doing it. This is just the natural state of how you're going to move. Versus active transport would be like this cat trying to climb up this slick slide. When you try to climb up the slide, you have to put in significant effort to get it to work. So looking at passive transport, we really call passive transport diffusion. All passive transport is effectively some type of diffusion. And diffusion is just the movement of particles from a high to a low concentration until equilibrium is reached. So everything that we've been saying is just natural, we define as diffusion. So when we're talking about molecules or ions or whatever it might be, the movement from high to low is diffusion. So if you take a droplet of food dye, put it in water, it has a high concentration where you originally put it, and it eventually spreads out from high to low until it makes the whole container blue. In terms of the membrane, it usually means we have tons of something on one side of the membrane and little to none on the other. And so those molecules and particles will move until there's an equal amount on both sides. So usually when we talk about diffusion, we're talking about a substance diffusing down its own gradient. If I were to put blue and red dye in here at the same time, the blue dye is not going to wait for the red dye to diffuse and then fill in the gaps. The red dye doesn't care what the blue dye is doing either. They will diffuse down their own gradients. Their idea is there's blue here, there's not blue over there, I'm going to go where there's not blue. Same thing with the red. The red only cares about the red. Now, there is one particular case where that's not really true, and it involves water. So when we have things dissolved in water, that makes it a little bit trickier, and we're going to talk about that later when we do a video on osmosis. So when we talk about diffusion, particles and molecules as they diffuse will follow Fick's law of diffusion. This tells us that the rate or the speed at which the diffusion is occurring, it's going to depend on a few things. One is the magnitude of the concentration gradient. If there's, 49% stuff on one side and 51% on the other side of the membrane, the rate of diffusion from 51 to 49 is going to be very slow because there's not a lot of potential energy, not a lot of pressure encouraging that movement. So it's going to just be a little bit slower. Versus if you have 100 and 0, that is a huge potential to get those molecules to move. And then it's also going to depend on the distance across which they have to diffuse. We'll talk about some examples when we see each other in person, but just know that if I have to walk across a room and the room is 3 feet across, I can do that much faster than if the room is 100 feet across. So the distance that you have to diffuse for molecules affects the rate. And then also the surface area. So cells are not visible to the naked eye for a reason, because they are limited to how much surface area to volume ratio they can have. So the cell membrane versus the interior have to be balanced, because this is how the cell gets in food and gets rid of waste through the membrane. So if the cell were to suddenly become really, really big, that would be great for diffusion, because now I have more areas for the particles to diffuse. If I make the cell impossibly small, I have less membrane and fewer areas for things to cross. A better example would be, would it be easier for all of us to run out of the classroom if the classroom were to suddenly burst into flames, if we only had a little doggy door for all of us to get out of, or if we had 5 large French doors that we could all run out of, or even a giant warehouse door that we could open up? That's the idea. The surface area, when we talk about particle movement, is important because the particles are moving across the surface area. Now, active transport is going against the flow. So this is where we want to move things against the concentration gradient. Instead of saying, hey, there's a lot on this side and a little here, I'm going to move down. It's saying, I want to take this and move it up to where there's more of it. We do this on purpose many times in the cell to create concentration gradients that we can capitalize on later. So when we do this, though, it's going to require the direct input of energy because we are fighting entropy. We are creating order out of chaos. So we do this, as I said, to create gradients on purpose, so that way when we're ready, we can open up a channel and allow all of this gradient that we've built up to flow through. And as we just learned with fixed rate of diffusion, that's going to be a lot of potential that we can capitalize on. So as that potential energy is converted into kinetic energy with particle movement or electricity through charged particle movement, the cell actually has ways of capturing that kinetic energy or electrical energy and converting it into usable energy. So if we're talking about bacteria, they will move hydrogen ions to one side of the membrane and then let them flow through, and that can power their flagella, their little tail, so they can swim around. We use it the same way that those hydrogen ions, we use it to spin a turbine in an enzyme called ATP synthase to create ATP, or energy molecules for the cell, which we'll get into later. Now, there's also this other form of active transport that we're just going to get through really quickly because we're not going to focus on it too much in this class. We can use active transport for bulk transport. What if I just want to take tons of molecules from the environment? What if I'm a white blood cell and there is a bacteria that I just want to swallow up and destroy? What if I have a very, very large molecule that cannot fit through any of my proteins? What do I do then? Well, that's where we have this active transport in bulk. So when that happens, we can either do an exocytosis or endocytosis. So let's get into the difference between those two. For exocytosis, this is where we have stuff inside the cell that we want to push out of the cell. So we create a little vesicle. This is like a tiny little membrane package, and we package whatever we want in there, move it up to the membrane, and then release it from the cell. The membrane of the vesicle will merge with the membrane of the cell membrane, and whatever's inside will get released. So we often use this for secretion. This is where we create specific things like hormones, and we use those to communicate with other cells in the body. So we need a way to get those hormones out, and this is how we do it. We also use it to get rid of waste, so we can capture a lot of waste in our lysosomes when we digest food. And instead of releasing them into the cell where they can harm the cell, we keep them in the vesicle and then have it merge and release the waste into the external environment. Now, exocytosis also is how we take neurotransmitters and release them from our neurons. So this right here is a neuron, the end of a neuron, one of our brain cells. And inside are little vesicles filled with neurotransmitters. And when we have a requirement for those neurotransmitters, we send an electrical signal down the nerve itself that causes all of these vesicles to move towards the membrane and release their neurotransmitters. Those neurotransmitters then attach to membranes on a different cell and continue the electrical signal down that next cell. That's how we kind of have thoughts. So that's one cool example. Then we also can do the opposite. We can do endocytosis, where we bring material into the cell. So think exocytosis, we're exiting something out of the cell. Endocytosis sounds like into, we're bringing something into the cell. So what usually happens, there's 3 different versions of this. Usually the membrane somehow curves inward and releases a vesicle with something trapped inside. Think of it as like reverse blowing a bubble. In this case, There's phagocytosis, where the membrane actually pushes out to surround whatever it wants to take in, and then it will trap that inside the vesicle. This is usually done to eat. So cells are usually grabbing whatever is out there for food. There is pinocytosis. This is where the cell just takes in liquid from the external environment, and if things get trapped in there, oh well, that wasn't really the point. The point was mainly just to take in more fluid. And then there is receptor-mediated endocytosis. This is where we only want specific things to come in. Only if we have, stars and diamonds, I only want stars to come into the cell. So I would need a protein on the surface that can attach to stars only. And when that happens, then it will start this process of this bubble coming into the cell and pinching off. And now I have a bunch of stars inside a vesicle, and I can do whatever I want with those stars. So that is it. We're going to do one more video in this set on the membrane about osmosis, and then we'd be done.